One of the things we could do with the model is to create diabetic flies that also have cancer. And Mount Sinai had done initial studies and published on it that patients that have diabetes should get completely different drug combinations than patients that just have cancer alone. And that's something that next year or 2 years is something we're going to pursue as well. And even for patients today, commercially, we can create a diabetic fly for them with their cancer.
That's absolutely wild. Welcome to Target Cancer Podcast. My name is Dr. Sanjay Janeja, also known as the Onc Doc online. I'm a hematologist medical oncologist, but more than anything else, I love having these kind of conversations. And in this specific case, I learned about something that just blew my mind. It really is one of the most encouraging things on how and where cancer is going. And for that reason, I couldn't be more excited and humbled to have Laura Towart, who has a background basically in ovarian health and empowering women, but then took a big transition and founded and is the CEO of Vivian Therapeutics. And in this thing, it's gonna blow your mind. They're basically engineering models of your exact kind of cancer type so that they could precisely and in a holistic, included way, see what your cancer is sensitive to. So Laura, the crazy thing that you were sharing with me when we were on our call, which again, I've just been talking about a lot, is you're doing something where you're looking at, targeted therapies, right? Molecular therapies, precision therapy that we talk about all the time on the podcast. And we use evidence, basically on data at first, sometimes in animal models, and then we do trials. But what you're looking at is, okay, yes, targets are very, relevant, but is there kind of more complexity? The answer is yes, when it comes to all kinds of other things that go into these targets and downstream and upstream. And so you have a model where you basically re-engineer those kind of mutations that are unique to somebody's personal tumor, right? And then you basically expose it to multiple things to see, is it in fact in real life sensitive or not?
That's exactly right. So the current standard of care is really looking at single agents and single mutations. And we realize that now that tumors are much more complex and there's multiple mutations driving tumor growth at any time, and other interactions with the genetics as well. But just to start with the genetics, there's not really any model that we have today that can really replicate the true complexity of a patient's cancer. And so our model, the fruit fly, we're able to quickly and efficiently engineer and recreate complex tumors in the same tissue where the patient would have the tumor, and then use those what we call avatars for high throughput screening and therapeutics discovery.
So when you say that you re-engineer the tumor, like basically what we're talking about is when we get neogenomic sequencing, we've talked about that a lot on our podcast, which is like NGS or sequence your tumor and see what the targets are. You get a whole bunch of mutations. You get ones that are clinically relevant in the sense of like targetable. So you'll get like targetable actionable mutations, which means, hey, we have therapy for these things. Then you get like relevance that don't necessarily mean that there's a target, but they can help us basically say, hey, these are kind of known escape mechanisms, why something may be resistant to another treatment that you would give in a general sense, or why something may be sensitive. Even though it can't be specifically targeted, we just know that there is a significance there when it comes to treatment response. But then you have a whole bunch of other things that are just like we're learning about, we know, maybe we don't know. How many of that entire DNA, or basically the blueprint, how many of those features on that blueprint are being coded into these mosquitoes? And is it an organ that's being coded into these fruit flies? Is it an organ in the fruit fly? Is it the fruit fly itself that's like, behaving as the organ? Kind of dumb it down for us, really.
Sure. So it starts off with the patient going to their oncologist, getting a tumor biopsy and a blood sample. And then we coordinate whole exome sequencing of the tumor and the blood. We do the sequencing of the tumor at a very deep depth so that we're resolving all of the tumor subclones, getting a full picture of kind of mutational complexity of that patient's cancer. And then we have a proprietary method that was first developed at Mount Sinai Medical Center and then further elaborated on at the company, which enables us to really interrogate or read that tumor exome and identify the mutations that are, one, driving the tumorigenesis, but then also what we call passenger mutations that are kind of hopping along and helping with things like vasculature development for the tumor, scaffolding, and things like that. And so I would say Mount Sinai and the technology that we use was one of the first to really start to think about how those factors play a role in the development of resistance mechanisms. And so we do integrate all of that into our models. We also look at copy number variations, and we're engineering the tumor to develop in the tissue corresponding to where the patient has the tumor. For example, for colorectal cancer, we model the tumors in the lowermost portion of the fly gut. And so the tumor is growing, it's interacting with other organ systems, it's interacting with vasculature, nerves, immune cells. There's somewhat of the same kind of micro environment that you would expect to have in and microbiome as in a patient. Similar, not the same. There's limitations in each model, but what we love about our method is that we're really able to replicate the complexity and in an organ specific way. Because with things like PDX mice, you're creating kind of subcutaneous tumors. We could engineer up to 20 mutations. So very complex
See, that's
tumors.
wild because, if you take a lot of our podcasts put together, like Siddhartha Mukherjee, right, Pulitzer Prize winner of enroll maladies, and what he was talking about with pancreatic cancer, and as well as the other ones with immune system, what you're saying is, where we talked in Sid's podcast about how you can basically tumors have a bunch of mechanisms to recruit your own cells locally to kind of shield or wall it off from the system or basically deflect immunity. We talked about how colorectal cancers and renal cell cancers have little properties to recruit blood vessel growth because that's their way for pathogenesis. They basically need that fuel and gas to either grow or to like hop away, which is why we use things that block blood vessel growth. But for the most part, like you told me on the call, which at first I scrunched my eyebrows because I was like, I don't want to believe that. And I'm like, yeah, you're kind of right. You're just attacking one thing in these instances that say, hey, let me just, ablate that one mechanism. So what y'all are doing, it's a Louisiana phrase, I don't know if you're familiar, but the phrase y'all, but what y'all are doing is saying, hey, let's just like do the best we can. You can't just put them into humans, obviously, that would be terrible. But how can we see all of those interplays and dynamics that go into something? Like, people don't know with colon cancer. They're like, why does it matter if it's right or left? I had a TikTok comment that this is like getting so ridiculous, like medical, traditional medicine wants us to believe this and that. But embryologically, meaning like when you're growing, they actually come from different areas, which is why right-sided behave differently than left. I actually made a video on that recently, why some of our treatments work and don't work depending on which side. So you said, let's do that. I mean, if that's the way it's behaving, let's do the closest thing we can to basically having all of those factors. And then what do you do after that? So you put those up to 20 mutations that the cancer has that you believe makes it cancer, makes it malignant, makes it pathological. All those terms mean this thing ain't doing what it's supposed to. What do you do when you've now created this sinister venom from Spider-Man kind of thing into these fruit flies?
So we expand the fly population, and for patients, we usually screen at least 500,000 flies. So we call this the Avatar Army. And you can imagine we have all of these flies, and we can then use them for high throughput drug screening. So we screen the whole FDA-approved library, or in Europe, the EMA-approved drug library. And the concept that came from Mount Sinai is like, we don't know what is going to work in these flies. We're not going to presume to know. Let's just throw the kitchen sink at it. And so I really love this unbiased approach to mass screening. And that's really what
And
our
what
name comes from.
constitutes that kitchen sink?
So basically, all of the drugs that the FDA makes available. So there's about 2,000 drugs that have ever been approved for anything, and we screen them alone and in combinations. And so over time, every time we do a screen, we don't screen 2,000, to the third permutation. We've generated intelligence, so we know now for a specific mutational profile what drug therapies are likely to work, and then we start experimenting with combinations. And so we're with each patient screen we do, we identify many novel drug combinations, usually a cancer drug plus one or two non-cancer drugs, which makes it treatment overall less toxic. And then the combination itself is quite affordable because we're looking at really small molecules and really interesting drugs. We get combinations like a MEK inhibitor with anti-asthmatic or an antihypertensive or metformin or very unique therapies that somehow are working to target that patient's unique mutational profile.
That is so wild. So if I understood you correctly, we've talked about multiple times on podcasts, how there's not going to be a cure for cancer. It's going to be cures like for cancer. Basically, it's going to be a cocktail because you have to block all the mechanisms and pathways. It's almost insulting to say one drug for something that's so challenging across the world is just going to fix it all, right? But what's really crazy about what you're saying in a good way is that, where one, we know certain targeted therapies for whatever reason, about 2 to 3% of the time work in a tumor where we're just like, bro, this has no reason. Why, I don't know, but they're getting a response from this targeted therapy. That happens. We know that happens. The second thing, though, that you're doing when you say FDA approved, and this has never been talked about on our podcast, you're saying you put in a cocktail or exposure of FDA approved medications and treatments that are not necessarily cancer treatments, but that have metabolic or physiologic relevance. Because we know, and I always say this, cancer cells were your regular cell. They have all the toolboxes of your regular cells. So if, beta blockers or inhalers for asthma or metformin, that it's not like they just hone out and go to the glucose thing or the lung thing. They basically have all kind of pathways where they have actions, and we know those pathways have to have a relevance in the growth of this cell because again, it was your normal cell. So what if this
Yeah.
thing that the bronchospasm inhaler, like if you block that, does it somehow, is that an escape mechanism nobody's even looking at? And that to me is absolutely wild. And you're finding success in a way. Like it's the tailored of all tailored ways of a cocktail on your physiologic cell. And I bet if you were to just apply that and say, hey, can I steal yours? I have colon cancer too. It's just not going to work that way, right? Just like some people get asthma and some don't. Some people Tylenol fix their pain. Other people like me, I'm like, how is it placebo? We all have different physiologies in our or acetaminophen, sorry, in our cellular makeup, and that's what you're looking at. Whatever your proclivities were since day one, how do they have an interplay with the cancer mutations that you have? And can we just shotgun an approach of therapies and see if it's somehow effective? Because we know that all these cascades add up, but not many people are looking into that. And if you just did mini studies on each one, it would take forever and be very costly. But two, like it wouldn't apply to just that one target. That's your whole concept. Instead, you need to do all of this for that specific cancer because you need to see how they all play together.
And it's really something we could only do in the fruit fly because we have such little animals. We're screening, millions, and we're able to do it quickly and cheaply. Well, one, we have a century worth of kind of genetic toolkits that have been developed to be able to manipulate the fly genome, and we can't really do that with any other organism. So it's really a very finely tuned system for drug therapeutic discovery and also development.
That's crazy. I mean, I keep trying to think of a metaphor to really like drive home how wild this is. And a lot of people say like, oh, I tried melatonin 5, 10, 15, and it didn't work, right? Melatonin like, we use for sleep here. I don't know what you use in London, probably something fancier. But then I tell patients, I'm like, actually 0.5 could work for you or 1, and it's not really weight and all this stuff. It's just you. And that's why the dosing is so challenging. And if you have more, it may not be working. But that's a very one-dimensional example, right? And then you can talk about, I was thinking about beverages with alcohol, like everyone, an old-fashioned, what kind of bitters do you use? Or a martini, is it gin, is it vodka, is it dry, is it not? Like everyone has their low preferences, and that's maybe a 1.5 dimension. But we're talking about cellular and physiology and everything that, again, you had innate, meaning somatic, meaning the stuff you were born with for your cell that's unique to you, properties, plus you have the tumor properties. But the coding you're doing in the fruit fly, that's only including the somatic, meaning the mutations that are in the cancer cells themselves. Do you apply something from what's called germline mutations in your normal cells or genetics also into that fruit fly?
We do whole exome sequencing of the tumor and the blood. We subtract a lot of the germline unless it's something that is very relevant that we should model.
Like
But
BRCA.
usually we try to subtract, yeah, so that we're really just getting what's driving that tumor. What to add even more complexity onto this, one of the things we could do with the model is to create diabetic flies that also have cancer. And Mount Sinai had done initial studies and published on it that that have diabetes should get completely different drug combinations than patients that just have cancer alone. And that's something
Oh yeah.
that next year or 2 years is something we're going to pursue as well. And even for patients today, commercially, we can create a diabetic fly for them with their cancer.
That's absolutely wild. And that's where the challenge is like, hey, how come we haven't cured that cancer yet? We cured polio and all that stuff. Like, why not? Are we holding out for money? That's because not only is the cancer type unique, versus colon one person versus the other, but also right and left, and also what the status of your immune system is, and also what your physiologic regulation for diabetes is. That's the humility, if anything, that we are constantly, regularly uncovering. I had an amazing podcast with Sandeep Patel at UC San Diego, where I found like they're looking at stuff and theorizing that even pancreatic treatments may or may not be effective because of the microbiome or bacteria that's around. Like some of the bacteria either metabolizes the treatments quickly or not. So you can really whittle down. And in an elite world, and I think this, if it seems like it's working, you keep going, then you introduce those bacterial species into the gut. And the whole concept is, I love the word avatar. Did you come up with that? That's
Epigenetic.
genius. It's create an avatar as close to yourself as you can.
Yeah, and there's so many layers. There's epigenetics, the microbiome. There's you can go into so many degrees of personalization. We're taking off kind of a big bite with what we're doing now, but in time we can layer and start to explore some of those things. Actually, with Imperial Medical Center here in London, we're doing a clinical study now that's integrating the patient's microbiome. So we'll get some preliminary data on that as well. But yeah, it's really exciting.
Is there, so in addition to like FDA approved medications that are, for other things like again, metformin, diabetes, asthma, etc., are there any models, or are y'all looking into things where you're introducing things like, I get a lot of questions on turmeric or ginger or CBD, marijuana, THC, all the things that are kind of popularized based on some limited data. Is that part of any of the cocktails yet, or?
So I think we have some natural compounds, but we really start off with the focus on the FDA approved library. If a patient or an oncologist had a particular interest, we could always add it. So that's one of the kind of the beauties of kind of doing something so personalized. If the oncologist was thinking, hey, I might not want to go, fully off piste and do something like a novel drug combination, but for my patient's next line of therapy, I'm thinking of A, B, or C. We can test A, B, or C and kind of rank them by efficacy. And then, for example, we're working with a clinic in Switzerland that is studying mistletoe therapy. I don't know if you've heard of it, but it's a locally active thing that they've had very good success stories.
I think one of the biggest points here that I don't know that we've highlighted enough in previous podcasts is, we talk about precision and how, but we don't build on that. And one of the points and concepts here is in 2023, we know, don't let anyone tell you otherwise, that in humility, again, it's just such a humbling feel. It's like Carl Sagan that says, like, astronomy is a humbling science or space, and I feel the same way about medicine, especially about cancer. We know that there are regimens that work that are so random to us that we just don't know like why, but they work on the cancer. Like in our own humble understanding, we're like, how does this mechanism play a role? But if you don't have it, that other treatment doesn't work in the cancer. And that you're taking that philosophy, that humility, and saying, we already have plenty of evidence of this. Let's see like how that relates not just to multiple cancer treatments and targeted therapies, but also, again, other things we know plays a part in cellular physiology. For the same reason, that's why I'm actually super happy and proud that because of XYours, which is a platform that queries all the basic case reports on random things, so like your cocktail is designed for that person, but if there's studies and somebody says, hey, I used this thing for melanoma, but I also used it for lung and it worked in this renal cell cancer, like I've had 3 patients in the last probably 6 weeks where it was like 5th or 6th line, been taking care of them for years, stage 4, good performance status, and there's literally nothing left in FDA approval. There were regimens that are medications I'm very comfortable with because I use them in other tumor types solo. And they gave me the case reports. I had no problem with insurance because I gave the case reports XYours gave me, which is a free service for doctors and for patients. And they all have stable disease. Like, I mean, those 3. Like, I've had it not work too. But to think that, like, now 2 or 3 months down the line, they'd be progressing, and there's no way, as a community oncologist or even academic, why you would think those would work. But somebody did it somewhere. Maybe it was based on a fruit fly similar thing from Mount Sinai or wherever. But that, I think, concept needs to really kind of disseminate in our world because, again, it just gives you the humility of how complicated this is.
Exactly. And it's something that we're really tackling now because a lot of the other companies that are out there are really helping oncologists to decide between different standard of care therapies, and not really having kind of the evidence to be able to say, we've done this in a model and the model is telling us this, which, because even though we're working with fruit flies, they are animals. And so we have animal data that's, gives information about dosing, efficacy, toxicity, and we're able to really generate these novel cocktails that hopefully will become more and more integrated into care.
So if somebody wanted to do this, they're listening. Like, number one, what line or where in their cancer diagnosis should they do it? I'm going to give you a couple questions. You're obviously ridiculously bright, so I know you can remember them all in order and say them backwards. I'm just kidding.
Okay.
But number one, like, where in the line should they do it? First line really early on, should they do it later? Number two, should someone stage 1, 2, or 3 be considering this? I'm going to go ahead and say probably not, because obviously there's obviously very well approved strategies, especially if you find the 5-year survival's 80-90%. You want to elect for something like that. So I assume it's stage 4. And then 3, how long is the turnaround? So how late in their disease course, how late is the turnaround? And it sounds like there's not much difficulty getting the medications. You could even pull up a third-party app to get the code for like an asthma medication and stuff, because for whatever reason it works. So sorry, that was actually too much for me to remember. How long does
No,
it take?
that's
When in their disease course should they do this?
Sure. So I'd say Mount Sinai started a clinical trial in 2015. The precondition for entry into the trial was patients had to be kind of end stage or stage 4, had failed previous therapies and have no other options. Out of those patients, it was a very limited number. 3 patients had outstanding success, and two published case studies came out of it. Here in London, we've been working with commercial patients from around the world. And we've also had some really standout patient survival stories. And we're starting to do a clinical study at Imperial here in London. And the idea is to start to treat patients earlier. So the patients were enrolled in the study upon diagnosis. They've had whole genome sequencing, which is just part of the grant. And we've built flies for them. We've done the screening. And now the oncologists are deciding when to integrate this into care, with the idea that it could be second or third line, which is really exciting. So we'll get some data to really be able to suggest, really improved outcomes from the start. Patients that come to us commercially, it really depends. It's oncologists that work with us that are comfortable with the combinations that we identify. We'll have the discussion with their patients, and it's really up to the patient and the oncologist when they begin. For patients that are going to get the personal discovery process, which is us creating a personalized fly for them for therapeutics discovery and screening, that could take up to 6 months. And for those patients, we really hope that they would start earlier so that they're not really pressured. We're not pressured to get them a recommendation when they need it, when standard of care starts to fail. And then with our new product, TuMatch is now available for colorectal cancer. And it's based upon all of the data that we've generated over years linked with patient's mutational profile and the appropriate treatment recommendation. So that's pretty instant. So that's where we're going in the
You didn't tell
future,
me
is
about
for all
that. Is that a new development?
yeah, so we've just launched TuMatch. So TuMatch is for colorectal cancer only at this point, but it's in development for other GI cancers, lung cancer, pancreatic cancer. And it basically enables us to go straight from whole exome sequence to treatment recommendation.
That is fantastic. So basically, like, if I don't have, so one, you say, 6 months if you do it just, purely like based on your mutations and everything. I like that, because somebody may hear 6 months, like, when you have stage 4, first of all stage 4s are treated differently and mean different things. But secondly, usually first lines in the major cancers like breast, lung, usually colorectal, like 6 months is hopefully like within still when your first line is working. So you're getting it up front, but then like especially in those cases where like small cell lung cancer, unfortunately, there's just not a lot after platinum therapy. And it's always hard for me to treat because the odds just aren't as good. So you want something to hopefully like have, in the queue. But some of these other
It's
tumors,
like
that's
an
more than
insurance
enough time
policy.
to be able to,
We call it like an insurance policy. So you kind of
yeah,
invest in it when you don't need it for when you actually will need it.
right.
Hopefully you
And
won't.
that's plenty of
You
lead
need it,
time.
it's there.
Right.
Yep.
Usually has plenty of lead time, like, with most first lines, fortunately, when it comes to the major tumor types, because of how long first lines generally work. I mean, even pancreatic, if you can use Folfoxiri or Gemabraxane, like, that you hope that you could, comfortably make it, or maybe be progressing, and then have the data back. But the two match is super exciting, because I do have patients where I'm like, I don't have that time or the finances. Obviously, like in an ideal world, you just offer it, for free to everybody. I hope. I know there's some good ones out there in the cancer industry, including on oncology, because you have to really. I think part of it is just admitting like it is a for business. Like I and you, like it seems like-minded in our just kind of like what we want to do for people and helping people. But you also have to accept the world is what it is. So we would do that for free
We
if we could. But it sounds like with
Yep.
two match, you're trying to at least get there sooner by saying, hey, these are the things that look very similar to your right-sided colorectal cancer, that's stage 4. Like, so a lot of things similar. And here's the mutational profiles, and this was some random cocktail that seemed to work on this patient. And if I'm in a bind and I don't have 6 months, I'm just delighted to hear that that's something that is also available now, and hopefully we'll see more tumor types as they come.
Yeah, well, it's just as good as the personal discovery process because basically what we've done is go into large data sets, large databases like Genomics England or the NIH, and we've stratified patients by mutational profile. And for example, for colorectal cancer, there's something like 450 unique mutational profiles that we've seen and that have been registered. And so we've gone through and we've started building avatars to match the most prevalent of those. And that's how TuMatch was built. So there's about
And what's the
100
what's the turnaround time on that?
so that's basic. That's TuMatch. So we've gone through matched. We built
Right.
mutations. We built avatars to match all of the most common mutational profiles, done the drug screening, and identified therapies. And that's the foundation for TuMatch. So that an incoming patient is actually matched by pretty precisely to either the exact mutations of a fly that we built and screened, or to there it's matched to a cluster by a machine learning algorithm that puts that mutational profile paired with another mutational cluster.
So really, like for that one, like other than outside of the time it would take to get the mutations, like the genomic sequencing with
Yeah.
Keras and some of the other things we use in the States, neogenomics, etc. Tempest, you outside of that time, when I have that information, it sounds like it'd just be a day or two to be able to see the cocktail if we use TuMatch.
Yep, it's pretty immediate. And so we're developing it as a SaaS tool, and we'll go through the software as a medical device regulatory process, which we've now started.
Wow, I'm going to get off this call and just query immediately my colorectals in like a third or fourth line. I mean, this is huge.
Okay.
So I guess you were just saving for that one for like a little like, and here's the thing on the podcast that we didn't talk about. So this is talk about previously. That's amazing. And so okay, what are some of the tumor types? I know we're both you're a very busy entrepreneur person, like doing everything. What are some of the tumor types, if any, that do seem to have more success, I guess, than others? Like, are there some like, yeah, we try, but they're just a little more stubborn, like really low grade lymphomas or something? Or is there some tumor type that's, I assume, probably different than your major ones? And are there some that it works really well in? That's number one. And number two, share a couple of success stories on like that stage four, like how long did this cocktail work? Are there people that are alive like a year or two later and that kind of thing?
So we licensed the technology in partnership with Mount Sinai, and Mount Sinai set off to really focus on colorectal cancer. And they chose colorectal cancer, one, because it's complex and hard to treat, and two, because most patients don't respond to immunotherapy. And so there's a big unmet need. And so we really focus on colorectal cancer, and we've had great success with it. I'd say the model is very good for rare genetic cancers, anything that's really genetically driven, not so much anything that has a strong hormonal component. So we kind of avoid breast cancers aside from triple negative. Um, we've done a prostate. It really depends on the markers that we identify.
Oh, that makes sense.
But yeah, but there's things like, of course, we can't do osteosarcomas. The flies don't have bones, and we try to find, we try to do the models in the exact tissue type that the patient has the tumor. Sometimes there's not a direct correlation. For example, for lung cancer, we build the model, and there's like a kind of a trachea type organ in the fly that permits for transfer of gases, gas exchange. So we do the modeling there. So it's not exact, but it's a pretty good enough model.
Yeah, and for the record, when you say, I know you know this, but when you say genetically driven, you don't mean like I want everyone else to know this. It's just it keeps getting misunderstood. Genetic does not mean that you have a gene that your parents gave you. By genetic, all you're saying is, hey, that gene that was a normal gene that has nothing to do with inheritance or BRCA, that gene that switched that we know turns on or tells cells to grow in all of our cells got stuck. That's the stuff that you're just like, ooh, like that's juicy stuff, which is a lot of driver mutation stuff in colorectal and all. And the hormone stuff is different because that is like we know that they're likely a consequence of basically being fed something that really helps them drive forward. And that makes sense why you don't necessarily like, need to, I would even guess, model that in fruit fly because one of the main mechanisms is that hormone.
Yeah, the other thing is we look to see if a patient what their microsatellite instability is. So we can't
I was
work
going to
with
ask that.
patients that yeah, we can't work with patients that are MSI high because obviously they're constantly developing new mutations. By the time we've created a model and screened it, their cancer might look totally different. So we only work with lower stable. It's also good because patients that are MSI high can be candidates for immunotherapy. So those
Right.
patients should go on that track. Ones that are not could go down our lane.
Yeah, and that's a great point you brought up because I feel, I was like hammer firing questions because we were on a call so short, like 6 months ago when we talked about it, and they were the hard pressing ones, and I'm like, well, how do you account for the mutational, colony that was like selected for with a progression and stuff like that, which and I'm now saying it's not necessarily progression, it's a persistence of a tumor that was there that you just are seeing grow and everything else dies. But that's a great point. So that's why you're saying like, hey, the ones that have mutational variability, obviously if we code it and 6 months later it's those 20 genes, like your new tumor like may be different. So when you go like lower grade or more stable, that means the mechanism is being replicated or duplicated in a carbon copy, hopefully more than, much more than these other ones, which is a very valid point. So, I mean, even presumably like slower tumors like renal cell carcinoma, I guess, if it's not immunogenic, as much on the profile as angiogenic. There's a previous podcast for anyone that wants to know the difference. And or the tumors that are like maybe lower grade lymphomas, those are good, I would guess, because they don't really change as much. They're just super stubborn to be able to treat. And then last question is the survival stories. You said you've had a couple of big
Oh,
standing cases.
yes.
There was two at Mount Sinai. Yeah, like what are we talking about? Like, hey, they got 3 or 4 months and everything, the disease was stable. Did we see shrinkage? Did we see actually things in a third, fourth line setting that just disappeared? And how long? Tell us about that.
So we've got a patient case study that's been published. It was published in 2019 that really kick-started us to license the technology and start offering it commercially immediately. This patient was 53 years old, man, lived in New York City, was a banker, had a nasty KRAS positive colorectal cancer, had 9 genetic alterations, identified and ultimately engineered in the fly, identified a novel therapeutic. It was a MEK inhibitor and a bisphosphonate. Patient was treated and realized a 45% reduction in his target lesions in 27 weeks and had, because it was a much kind of nicer cocktail than what he had been treated with before, had an improved health related quality of life, went back to work, was going to the gym, and had an 11-month extension of life. And during that period,
Wow.
it was family, and he was very pleased. So that was considered a great success. Another patient, also from the Mount Sinai trial, was a patient with adenoid cystic carcinoma. A novel 3-drug combination was identified. It was one cancer drug, 2 non-cancer drugs. Patient ultimately died of COVID, so he had an extreme extension of life, and who knows if he didn't get COVID, how long he would have lived. But also health related quality of life improvements there as well. So really exciting stuff.
That's amazing. And for people that don't know, like KRAS mutated, it's just not ideal. It's they're more aggressive. They don't respond to therapy as much. So that's huge. And it sounds like, because I have one patient I can think off the top of my head in the past few weeks that I am kind of getting in a bind with. It sounds like I can like hopefully hope, like that's a medical exact like science term, that that might be available in the two that new that what is the new platform called? Because if his profile
TuMatch.
matches, I have a two match where I could actually have a cocktail kind of why not, kind of concept.
Yeah, with TuMatch, so we liked for patients to give us whole exome sequence data, but we could also work, and we know patients sometimes don't have time, or maybe they don't have a biopsy sample existing and available. So if they've got a Tempus, a Foundation One, a Guardant, we can take a look at it and we can let the patient and the oncologist know if we have a match. So then once we have a match, then we can discuss payment and things like that. But just if you were curious if there is something available, we can
Yeah, so you don't
immediately.
waste your money. It's not like you pay it up front and then you hope and then you're like, oh man, I didn't even apply. So that's
Sorry.
that's really thoughtful. Yeah. Oh,
Yeah.
that's thoughtful. That's awesome. Okay, we've talked about so many exciting things that like I hope anyone hearing this one more than anything is humbled again by the challenges of like cancer and how like nebulous and enigmatic it is. But two, like very optimistic about really like where we're going and how things are just viewed differently thanks to people like you and Mount Sinai on saying like, hey, we know what the issues are. We know that there's a lot of interplay. We can do the interplay. Like let's like model it the best we can. So because of all that exciting stuff, I'm going to do something that's not as exciting in conversation, but important. Can you define for us a little bit when you say whole exome or RNA or DNA? Like give us the kind of 90-second to 3 minute, not even 3 minute basics on what those differences are. How would a patient know when they're oncologist and they ask for their sequencing or molecular? Like how would they? What are we talking about?
Sure. So basically, your genome is all of the genes that are encoded to make you you, including exons and introns. And so we do whole genome sequencing as well. And I'm usually a proponent of whole genome sequencing because I'm a scientist and I want to know exactly what all of the things that are there, even if we don't understand the whole genome today and can't make use of it. I'd like to collect that data so that as we start to uncover and learn more, we can go back and kind of restudy some of the patients that we've had in the past. So I
And
personally love to have,
that's again germline like genome stuff. Like I just want people to kind of compartmentalize. Like that's just what you're
well, we,
given. And yeah, well, I was
well,
just saying
we look
because that's why some people are like, I smoked my whole life and never got COPD and cancer. I don't believe it. It's like that is maybe very likely in part you don't have COPD or cancer because of your genome, like your sequencing and all these things that you as a scientist are trying to learn and say, oh, this is why. Like you got to find that stuff. It's not given to us in the Bible.
That's right. Well, we do whole genome sequencing of the tumor as well. So we really
Right.
dive deep into the tumor. But we realize that in practice today, we're not utilizing the genome as much as the exome is fine. Like it's enough. The exome is made up of exons, which are the coding regions. So you're just eliminating all of the non-coding regions, getting rid of that so we can just focus on kind of what we know and what we understand today. And
And
then
what
in
are we
terms of
losing with the with not having the introns?
so you kind of get rid of the introns. Introns are just the non-coding regions, so we don't need to take a look at those. They're not really involved in driving our therapies today. So the
They're not the
exons
ones
and
that
the
are involved in creating the proteins and the tools. Like they're not the blueprints for like the
exactly,
hammer, the nail, the screwdriver, right? Okay.
exactly. And then in terms of mutational panels, it's just looking at known mutations in genes that we know that are causative of different cancer types, and we know how to target those mutations. So companies have just made simplified panels, and we can work with that. There's more complexity to our method than you can really see in a mutational panel, but very good new mutational panels usually cover up quite a bit.
I'm so happy and inspired, but I'm also drained because it's just so exciting and so mind blowing and so all of these other things. And this is just you just walk and talk and breathe this every day. And I can't. I love that I get to speak with super people. So thank you. Is there anything you want to leave people with? I hope I know I found or one of us found each other on LinkedIn. You're there, so people can keep seeing what you're doing in science and medicine over your career. And then Vivian, obviously, therapeutics. We're going to put in the link. But is there something else that you just want to share that when you're having these conversations?
I just would hope that everyone that, I've personally dealt with cancer in my family, and I know when cancer is diagnosed, people, it's very worrying and for good reason. But there are new therapies out there, and, anyone who's interested in even learning more about what we do at Vivan and if we could potentially help them or a family member or friend, please reach out. We're really friendly and we like talking with people and trying to do the best we can to help. And then also same with oncologists and medical centers. We're very collaborative. We're really interested in pursuing some of these, the additional layers of personalization. We're really interested in forging new relationships and working together.
The good ones, fighting the good fight. We appreciate you so much, Laura. Thank you.
Great. Thank you so much. I really appreciate it. Thanks for having me.